Doing well, thank you.
Perfect, perfect. Okay, I think I can see more participation today. That make me much more happier.
Great.
Yeah. Okay. How are you doing? How are you doing? Everything is good. Perfect, perfect. Okay. So, few days ago, probably 2 days ago, I posted on eCampus a new assignment for you. And this time, something similar to what you did before about the identification of gram-negative or Enterobacteriaceae, some members from Enterobacteriaceae. But this time you are assigned Staphylococcus and Streptococcus species. So you know the genus, and I need from you to be capable of telling me the species based on the biochemical information that I sent you, okay? And I'd like to go through this and make it kind of more easy for you to clarify some issue about that. But before I go and start, do you have any question for me regarding the Enterobacteriaceae?
I do, Professor.
Yes, go ahead, please.
On each one of the postings, there were 3 exercises, 1, 2, and 3 for staph and strepto. Are we being assigned one of the 3 for each?
Yes, this is exactly, if you look at the announcement, you will see names, okay? So say, for example, Staphylococcus isolate number 1. And then you will see maybe like 6 or 7 students' names on this.
Okay.
So you need, first of all, to look for, search for your name in the list and see exactly what isolate you assigned for.
Oh gosh, I see it now. I just missed it. Okay,
Yeah,
thank
so you are responsible for identification of only one staph and one strep.
Okay, got it.
Just
I'm
one
sorry,
each, yeah,
I
one
missed
each.
that.
Similar to the Enterobacteriaceae, exactly.
Got it.
Okay, yeah, yeah.
Okay.
Okay, and for Enterobacteriaceae, if you have, anybody has any question for me? I know that some students still have some confusion about that assignment, and I'd like, hopefully they are like meeting with us today. So please, if you have any concern, share it with us so we can go through this.
If nobody has any questions, I just have a quick question on previous labs. I noticed that some of the labs haven't been graded yet that were turned in almost a couple weeks ago. Is that accurate, like the protozoa, fungi, and Kirby Bauer?
Yeah, you don't need to worry about the, as soon as you didn't receive any response back from me, that I received it. If when I read your email, I respond back that I received it. Okay, so again, I'm kind of a little bit behind.
Okay.
Because lots of assignments, lots of labs, different classes, stuff like that. So I'm kind of overwhelmed with numbers of assignments. But yeah, I promise, I assure you, you will be able to see more grades posted very soon. Yes.
Okay, thank you.
And after I finish everything, then I will look again, and if I see that like it was not graded yet for your name, then you will see an email from me, okay, and ask, maybe you sent it a couple of weeks ago, but I overlooked it, or maybe I didn't receive it at all. So we will be in touch. No worry about that.
Got it. Okay, thanks.
Yeah. Okay, so for Enterobacteriaceae, you have any question for me? Again, you were there are only 3 unknown, and each student is responsible for 1 unknown, okay? And as I mentioned before, the best thing to start with is a slide that show the lactose fermentation, because that lactose fermentation test can differentiate Enterobacteriaceae into 2 major groups. Those who can ferment lactose, because those lactose positive, and the way you will be able to tell by looking at the media for the lactose, and if you see the media turn from red to yellow, that tells you it is lactose positive, okay? And some of them also can accumulate gas, so they can produce gas and acid at the same time. And then after that, look for hydrogen sulfide formation from the SIM media. So use this chart, and that chart will direct you to the ice unknown that you are dealing with. It will be easy and very straightforward. And again, if you have any question for me, anything which is not clear, please email me. Don't hesitate to email me. Even I can do like a private Zoom meeting, so we can go through this together. Okay.
Thank you.
So now, yeah, any question for me?
No.
Staph. So now we are dealing with a completely different group of bacteria, and those are gram-positive. So remember, Enterobacteriaceae, the whole thing was gram-negative. Now we are dealing with gram-positive. Two major genera in gram-positive bacteria, and both are non-spore formers, so they cannot form any spores. Those are Staphylococcus and Streptococcus. Those are basically opportunistic pathogen, and they are everywhere. If you look at the human body, they are everywhere. You are in the skin, they are in your nostrils, they are on your throat. So they are very well associated with human. Opportunistic pathogen, meaning that if they get the chance and the environment, turn it to be like inductive for kind of pathogenicity, they will not hesitate and they will start attacking you without mercy. They are dangerous stuff. They are loaded with virulence factors. So for the staph lab, you have the genus Staphylococcus, and you're responsible to tell me the species that you have. Staphylococcus, those are mainly the arrangement. From the name, they are cocci, so the shape of the cells are cocci, spherical, and the arrangement, they are staph, like grape-like arrangement. There are gram-positive, so if you test for gram stain, they will take the blue color, indicating that they are gram-positive. They are facultative, uh, and aerobic, meaning they can grow in the presence or absence of oxygen. They have a very important thing, which is their ability to tolerate high salt, and we call this haloguric. There is a difference between haloguric and halophilic. Halophilic, they love salt, and if you deprive them from salt, they are not going to grow. Haloguric, on the other hand, they can tolerate high salt concentration. Staphylococcus can tolerate up to 15% salt in the media without affecting them. There are many species, as you can see, all those in red, and some of those are like saprophytes, meaning like Saprophyticus, they like to feed on dead stuff, and some are highly pathogenic, like Staphylococcus aureus. Okay, epidermolysis, for example, hemolyctus. So what is your job? Your job is to be able to identify the species, okay? So for this test or this lab, it is so important in your practical that is coming in a couple of weeks, practical 2-3 weeks, I believe, very close to the end, beginning of August. You need to know the media, exactly like Enterobacteriaceae. Then the name of the media, what is the media stands for, if there is any indicator, what is the name of the indicator, how it looks, if it is positive, how it looks, if it is negative, all those good stuff. You are going to do the same thing for Staphylococcus, okay? Staphylococcus, when you do catalase, they are catalase positive. It's like Enterobacteriaceae, so they are catalase positive. They are oxidase negative, okay? Oxidase negative. The first media that you need to know, and this is a very specific media for, we call it selective media for staph. We call it the MSA media, MSA, and MSA stands for mannitol salt agar. So the media, as you can see, it's red color. It has mannitol as the carbon, as the sugar mannitol, and it is red because it has a pH indicator, which is phenol red. That's why it is red. Okay, and what else? It has high salt concentration, so 7.5 salt. So it is very specific for staph. Okay, so if you have a staphylococcus grown in this media, you look for two things. First of all, the ability to grow and tolerate this high salt, and number two, their ability to ferment mannitol. So if they can ferment mannitol, the media is going to turn yellow, as you can see this. So here you have a staph which can grow at 7.5% salt, and also it can ferment mannitol. So this is the first media, and this is the media that you are going, basically, if in the real lab, we are isolating staph on this media, and the source. Can anybody tell me the source of the staph that you are going to isolate or um, like identify? What is the source? Do you get it from soil or from where?
Um,
Based on the lab.
the skin.
That's one thing, but the main source for staph for isolation will be from the nostrils, from the nose.
Okay.
Okay, nostril from the nose. So you have a cotton swab that you can go like maybe like a little bit more than an inch and try to take like sample from the internal part of the nostril and then spread it over the media and then incubate it. Uh, those kind of bacteria or facultative anaerobic, so they prefer low oxygen. So one way to grow them is to put them in like in a candle jar, a jar that has a burning candle, so it has less oxygen. This is the best environment for those bacteria to grow. Okay. So this is the first media that you need to be aware of: MSA media, mannitol, salt agar. The second kind of media is a Columbia nalidixic agar plates, or CNA plates. CNA plates, it is unique because it has two things. It has blood, sheep blood, and it has an antibiotic, nalidixic acid as antibiotic. So it is differential and selective. Nalidixic acid, most or all gram-negative bacteria are sensitive. So if you try to take an inoculum from the nostril and isolate, that will make sure that anything that appear on the plate will be gram-positive, because gram-negative will be completely out and removed because they cannot grow in this kind of media. And also it has blood, and you look for blood hemolysis, and this is important because I'm sharing with you my presentation. Can you see it, or I'm just talking from like and you're imagining what I'm talking about?
No, we can see it.
Great. Yeah, so if you look at hemolysis, generally speaking, bacteria can be divided into three groups based on the shape of the hemolytic zone around the colony. Alpha hemolytic bacteria, those cannot do complete hemolysis or lysis of red blood cell. Okay, so they are kind of grayish color, as you can see. Very little clear zone around them, not very clear, it's gray. We call those alpha. And there is complete hemolysis where the red blood cells completely lysed, and we call those the beta hemolytic group. Okay, and then there is another group, we call it gamma, and they are not hemolytic at all. So bacteria can be divided based on hemolysis into three groups: alpha, beta, and gamma, where beta has a complete hemolysis, alpha has partial hemolysis, gamma no hemolysis. Okay, so this is the media here, the Colombian nalidixic acid. It has blood and it has antibiotic. So you need to look for, after you grow your bacteria in the media here, you can also add another antibiotic. So the media is already has nalidixic acid to prevent gram-negative, and also you can add novobiocin, like a disc saturated with the antibiotic novobiocin while you are growing your organism. One important thing you need to do for this kind of assay to make sure you are spreading your inoculum very, very well to cover the whole surface. Don't leave any area without any inoculation, because you need to have a very complete, like the whole thing is covered completely, so you will be able to assess for the clearing zone. Okay, so some staphylococcus are sensitive to novobiocin, some are resistant. And the key here is the inhibition zone around the novobiocin disc. If the inhibition zone or the clearing area around the antibiotic is more than 17 millimeter in diameter, it tells you that your bacteria is sensitive to novobiocin. If it is less than 17 millimeter, that is an indication that your organism is resistant to novobiocin. Okay, so when I give you your isolate, you need to look for novobiocin because it's a very important feature for identification. Like here in this case, you look at this and you tell me, is it, what kind of hemolysis is this in your opinion? Is it alpha, beta, or gamma?
Just a second, I think it was the. I need to look at that screen again.
Yeah, so this
It
is,
was
we
the.
call this
Oh yeah.
complete lysis, right? Because very nice clear zone around the colony, as you can see. So it's not
Beta.
greenish. So
Beta
this is beta.
complete.
Yeah, this is beta, beta, okay? Somebody will tell me, oh, what is this? Is this alpha? No, it is once nice culture, but it is, you know, the incubation time was not enough, or maybe shadow of some light. So you can see that the colony kind of greenish a little bit. No, but this is picture. But the whole thing, it's a pure culture, and the kind of hemolysis is beta. And then the second thing that you need to look for is the ability to how they react with the antibiotic novobiocin, and by measuring the inhibition zone. So in this case, we measured it for you, say, and we give you an inhibition zone of 25 millimeter, and that indicate that this particular unknown is sensitive to novobiocin. So you have two things now. Here, you go back. So it can grow in high salt and it can ferment mannitol because the color change. Here, it is beta hemolytic and it is sensitive to novobiocin. So those are four different biochemical data that you need to gather. Then, based on also the ability to ferment lactose, it is very, very similar to the Enterobacteriaceae. The media has lactose sugar, and it has the pH indicator, which is phenol red. If there is any fermentation, it will give you a nice yellow color like that due to acidity, acid production, and gas accumulation. So your isolate in this case is fermenting lactose. Another important thing, the media, another kind of media called SM110. And also you need to know what is the purpose of this media. It is Staphylococcus medium 110, very, very similar to the mannitol salt agar that we discussed earlier. But it tests for the pigmentation. So some Staphylococcus, they have the ability to form or make pigments. In this case, like golden pigments, okay? This is a very, very characteristic feature for Staphylococcus aureus. Staphylococcus aureus, or aureus here golden, the name, because they can give this kind of nice golden color colonies on the plate. The media here is SM110. In the lab, also you can use SM110 to test for the ability of your unknown to hydrolyze gelatin. Okay? And the way you do this, after you grow your bacteria, you flood the plate with the saturated aqueous solution of ammonium sulfate and look for clearing zone around the colony. Okay. But for the purpose of this, we didn't mention anything whether they can hydrolyze gelatin or no. We said just are those kind of bacteria form pigment or no. And based on this figure, they are making pigments, okay, in the SM110 media. So how many media now we mentioned? We mentioned mannitol salt agar for isolation, okay, and mannitol fermentation. We mentioned the CNA media, Colombian nalidixic acid media for identification of the hemolysis and the sensitivity or resistant to novobiocin. And this is the third media, SM110 for pigment fermentation or pigmentation, okay? And then another test for identification. You have a
Professor,
question?
can I ask? Yes, I do.
Yes, yeah, go ahead.
On the previous slide, is there an alternative to the golden color?
Mm-hmm,
I'm looking
mm-hmm.
at my assignment, and mine looks like a pale yellow.
Like this, or kind of
No,
very, so it is negative. So it is
very
not
light.
pigmented. Yeah, it is not pigmented. It
But
should give you a very clear yellow pigmentation like that. And
well,
this is the only, yeah, go ahead.
I I can see the line perfectly, but it doesn't look like the picture you're showing me.
Yeah, this is a classic positive for pigment formation. Okay, so other bacteria, you will see them very similar to the surrounding. Okay, and those mean that they are not making any pigments.
So you can still see the streak if it's a different color. It's negative.
Yes, because this is
Okay.
growth. Yes, they
Okay.
growing. These are
Got it.
growing in the media, but if they accumulate, if they make pigments, they will show up very clear yellow, as you can see here. Okay, if it is kind of creamy like
Mm-hmm.
whitish color, a light yellow color, so they are not pigment for
Got it.
making. Yes.
Thank you.
Okay, and then another important thing now, you will start looking for some virulence factors, because we mentioned that staphylococcus, some species of staphylococcus are highly virulent, and some of those virulence factors like DNAs. DNAs is an enzyme that degrade DNA and hydrolyzing it and breaking it down to simple nucleotides. So the media here for DNA testing, you grow your bacteria, and as you can see, the media is kind of bluish color. And why it is blue? Because it has a dye, and that dye is methyl green. Methyl green, and it has also DNA. So you have DNA mixed with methyl green in the media, plus other carbon and nitrogen source. Okay, so if the bacteria is capable of hydrolyzing DNA because they have DNases, then you will see a very nice clear zone around the colony. Okay, and because the degradation of the methyl green binding DNA, so it will give you this very nice clear zone. This is one important experiment for to test for virulence factors like DNases. Another important virulence factor is going to be the coagulase. And the coagulase test, you basically take after you grow your bacteria, you can take a little inoculum and mix it with rabbit plasma. So the rabbit plasma is liquid and it has a fibrinogen on the plasma. So if the bacteria, if your staph is capable of coagulation and converting fibrinogen to fibrin, they make clots, okay? So what's going to happen? This fibrinogen will change from liquid to solid, okay? As you can see here, we are inverting the tube and it looks like, everything is coagulated. So if it is liquid, it will move all the way down here. But now you can see it's kind of coagulated, meaning that this particular unknown is coagulase plus, okay? So now you go to this very nice table. You know that you have a genus Staphylococcus, and you need to know the species. So like what we did for Enterobacteriaceae, we looked first at the lactose fermentation. Here we look for coagulase production. If they are they are like produce coagulase or plus, that will separate those very important species, areas and intermediates, from the rest. If yours is coagulase minus, then you are going to end up with those species here. Okay, so now you will be capable after that by looking for, if you are in the minus coagulase, look for the novobiocin resistance. Again, look for this diameter. If it is less than 17 millimeter, they are sensitive, and then you are down here. If they are more than if they are less than 17 millimeter, they are resistant, and you are here. If the inhibition zone is more than 17 millimeter, it's bigger, so they are sensitive, okay? And you are here. And then you go for other tests by looking at your presentation. This is for staff. And I will be more than happy to hear from you any question, okay? If you look at your presentation that I gave you, you will see at the end, this is like different kind of different species from Staphylococcus, and this is what I gave you, okay? So I give the class 3 different species. Some of you assigned Aries, some of you assigned Epidermoides, and some others assigned Saprophyticus. Okay, so you need to look for this kind of media here and this kind of biochemical testing and compare and see which one is your unknown. As soon as you identify this, you are doing the same thing like what you did for Enterobacteriaceae. You provide me with the ID sheet and then talk a little bit about that particular unknown and the importance or the significance, medical significance, and how it is, if there is any disease can be caused by this, and like how it is being treated.
Professor, I have a question on the genus
Yes.
Staphylococcus. At the junction of novobiocin resistant,
Mm-hmm.
um, if there is no zone of inhibition, it's resistant.
Definitely yes.
And that is that considered a plus or minus?
No, it's a plus.
Plus.
Yeah, it is resistant. It is resistant. So plus here means
Okay.
resistant to
Okay.
that antibiotic. Okay. Minus means it is sensitive to antibiotic.
Sensitive. Okay,
Yeah.
got it.
Okay. Like here, see, for sensitivity of novobiocin. So if you see no inhibition zone at all, so yours is resistant. Okay. If you see inhibition zone, you need to measure. If it is less than 17 millimeter, it is still resistant. If it is more than 17 millimeter, it will be sensitive.
Are we so if you use this chart and go to the positive or negative resistance, and as you go down the flow chart, are we going all the way to the bottom, or just are we stopping at the
Which one? Are you talking about this one or are you talking about which one?
the one you have on the screen right now,
No,
genus?
this one. Oh, sorry. So sorry.
Yeah.
Let me take this back because I need to stop sharing and
Yeah.
get you another one. I'm so sorry about that, man.
Yeah.
Sometimes I feel like I'm in class in the lab,
Uh-huh.
so I don't, I can't, I
So you're
can't.
going to show us another chart after
Yeah,
after we get to resistance?
yeah. Let me show
Oh,
you
okay.
this one.
Got it. Got it. Okay.
Yeah, this is what you have, right? The one that I
Mm-hmm.
have, this is a demonstration for when I teach in the lab. That is
Yes.
my presentation. This is what you have, right? So what
Yes.
you have here is very simple form. So you are dealing, the whole class assigned 3 different species, okay?
Mm-hmm.
So you need to look for your name and the isolate number or unknown number and work with it. again, look for hemolysis, look for the sensitivity to novobiosin. So here again, so novobiosin, you mentioned that if there is no inhibition zone at all, so you are resistant to novobiosin. If your inhibition zone is 17 millimeter or less, you still resistant. If your inhibition zone more than 17 millimeter in diameter, that is sensitive to novobiosin. Okay, look at the pigment production on the SM100 media. Okay, so if it is yellow versus kind of golden color versus whitish yellowish color. Okay, so that can differentiate easy between areas on the other two here. See, so what you are going to see, you are going to see a very, very simple assignment in front of you. Very few slides and very few, very limited species number. So that will make you like concentrating very well on the unknown that you have in hand. So it's not going to be as confusing as the enterobacteriaceae.
Yeah.
So this is for Staphylococcus. So I'd like to hear any question from you. So this is how they basically, it's like an inch, so you can take the cotton swab and try to rotate it. So you're going to collect, because they like to reside in the interior part of the nose like that. But remember, you have many other microflora, not only Staphylococcus. So other things are still there. And this is why you need to use the selective media first, which is the MSA media. It has high salt. Only Staphylococcus can tolerate high salt. The others will not. And if for any reason you have other that can tolerate high salt, say gram negative, then the second media, which is CNA media, it has nalidixic acid, and that will be enough to eliminate any gram negative. So you're going to deal only with gram positive.
Now, Professor, we talked about all of the tests, biochemical tests, but we did not talk about catalase, and that's one of the items listed on the lab sheet.
Okay, let me see here. You mean, what do you mean? You have it, right? You have
I didn't see
catalase.
anything in my.
You should have a catalase. You have a catalase, I believe.
I don't see it mentioned on my.
So what isolate number you have?
I am 3.
Okay, let me see what I have here. Let me do this. See why.
And I just wasn't sure if we, because in the yeah, it's not mentioned in the.
It is not mentioned in this table.
Yes, that table.
So it does mean that it is not really, for mainly all Staphylococcus, the genus itself is catalase positive. Okay. So this is, if I say catalase and put positive, 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 do you think that I will be able to use it for differentiation? If
No.
all species here that you are dealing with, those 3 species are catalase positive. So that test is not going to be enough for differentiation, right?
Yeah, okay.
So this is probably why you don't have it in your, but let me make sure about this. Bear with me for a few seconds here.
Oh, I guess I see it on slide 9, I guess. It's mentioned on that last bullet point.
Yes, it is there. Can you see it in slide 9?
Yes. And
It
if
is
it's
very clear.
bubbling like that, it's positive.
Yes, yes.
Got it. Okay.
Exactly. Yeah, you have it.
Thank
You should have it. Yeah.
Okay, thank you.
Okay. Okay, no problem. Anything for Staphylococcus before we jump to Streptococcus?
Well, I think we just almost did the lab right there, so thank you for that. Appreciate it.
No problem. Okay. Do I have anybody else with me? Or they left already the chatting room? Hello? Hello, hello.
Hello, sir.
Are you still
It's
there?
okay. I'm here, but
Oh,
it's okay. I muted, that's why.
oh, you didn't go to do some cooking or something quickly?
No, no, no. Already I'm done. I muted, that's why. I
Okay,
muted.
okay, no problem. Oh, you muted? Why? I didn't mute anybody. I did not. Yeah, yeah, I can see here. I don't know why.
I think
Can you
people
do it
just,
from your side? Because I can't even do it from my side.
yes, you can. I think just people do that because of background noise.
Ah, okay, okay. Okay, so I'm the one who did it. I didn't mute anybody. Okay, so now let's move to the second lab. And this one, let me close that one here. Okay. And let me share with you. Um, so the second lab, another important genus here is going to be Streptococcus. So I'm sharing with you this presentation here, okay? So can you see it?
Yes.
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 Yeah. So for Streptococcus and Staphylococcus, they are sharing some feature. They are kind of very, very close. Both are gram-positive. Both are cocci, spherical shape. Both are facultative anaerobic. Both are opportunistic human pathogen, non-spore former. Um, so the main one big difference between the two um, genera is the shape under the microscope. So I said that they are both gram-positive, but if you look at the cell arrangement, so they are cocci, Streptococcus, Staphylococcus, they are cocci, spherical, but the arrangement under a microscope is different. So Streptococcus like strip, making like a thread, making like the arrangement here, cells are attached to each other, making like short chain, sometimes a little bit longer chain of those cocci. So this is one feature to differentiate between staph and strep under the microscope. So staph cluster, grape-like. Strep is kind of like line like this, like thread like that, okay? This is one major difference. Also, if you grow them on the lab, you can use agar, blood agar plate, okay? But here it is not like mannitol salt agar, not like staph. Staph can tolerate high salt, strep can't, okay? This is another difference. So when you do just regular cheap blood agar, no salt, no mannitol, nothing, okay? So you isolate basic, what do you think the source for strep? So we know that the source for staph is going to be from the nostril. What about the strep? What do you think the best source to be able to get strep sample?
Let's see.
So the best,
Throat. Throat.
yes. Throat, yeah, you know, strep coming from throat, and this is when you have some kind of like pain in your throat or something like this, and you're running some high fever, you know, and the first thing they can do, they can, if you have kind of strep throat, you have strep infection. So what they do, they take like a cotton swab and they go inside your throat and try to take a sample and see if you have what strep and what kind, because again there are normal flora in your mouth. Strep, some species are kind of not harming a lot, but some of those species of strep, they are very dangerous pathogenic bacteria, okay? So when you take like a swab, a cotton swab, and you start like plating them and trying very much to get like a single colony separation, so you inoculate again, because they like staph. They prefer low oxygen concentration. So you put them in this candle jar. And when you do this, by the way, in the lab, You have a jar, you get a candle, you light the candle, and the candle is going to be like on like this for few seconds, maybe 30 seconds, 40 seconds, till most of the oxygen in the jar is going to be eliminated. Then it will be burned and it will be kind of off. So you can inoculate you put your plates inside the jar and close it tightly. So that will create less oxygen environment in the jar. They prefer this kind of condition. So when they,
Professor,
yes.
on the in the jar, candle jar, what do the does the smoke contaminate the jar and the and
If even if this is the case, if there is any kind of contamination, remember that your plate is covered. So
okay,
you inoculate your plate and you cover it with the cover like that. So you are preventing any kind of contamination. If there is any, you know, like any contamination in the jar, it will be hard for those bacteria to you know, like to fill in the plate because the plate is covered,
okay,
unless you have the plate uncovered, and this is dangerous because if you have this case, it will be definitely contaminated because this is not a very well contained environment, you know. Yeah,
hmm,
so
okay,
by covering it like that, you are preventing any kind of contamination. Yeah. So you will, if you do this, you will end up with all the microflora in your throat. Okay, and now it's your job. You have single colony, as you can see. Now your job is try very much to see if you have any strep. Okay, so you grow them in blood agar. If you go here in this table, okay, and look for hemolysis, again, some streptococcus, they are beta, some are alpha, and some are gamma. Okay, so when you grow your bacterium on the blood agar plate, you look for hemolysis. So this is exactly the same in a CNA media, exactly similar to the one that we use for staph. It inhibits, okay, remember here, you have blood agar if there is no nalidixic acid, so gram positive as well as gram negative will grow. But if you have a CNA plate, so only gram positive will grow, and then you will start look for the hemolysis. Okay, here if you end up with beta hemolysis like this, and some other bacteria that is kind of greenish color, so this is alpha hemolytic partially. Okay, so if you are really looking for streptococcus which are pathogenic, those are the beta group. So you can pick from those colony and you can use for isolation. Okay. So if you have incubation like that and you see this picture, then you can see that the majority of the colony that you have is beta hemolysis, beta hemolytic. So this is the first feature you are going to record, beta hemolytic. What else? The second thing is two different antibiotics that you are using for differentiation of different species of strep, two different kind of antibiotics. Here it shows you when you do like a very good bacterial lawn, this is bad. Why this is bad? Because the bacteria is not covering everything. Bacteria is concentrated like here, but you have still in the periphery area of inhibition, area of clearance. So if for any reason you put the antibiotic saturated disc in an area like that, it will give you false positive. Okay, because you already have no growth. So if you put it and see no inhibition, you see inhibition zone, oh, my bacteria is sensitive. In fact, no, it is not good result because the area itself was kind of no growth. So you need to make sure you have a plate covered like that. So if I gave you in your practical, I said 2 plates for antibiotic testing, which plate should be the right plate? Don't say I'm looking for good separation. You're not after separation here. You need really to have a very nice covering like that. Use two different kind of antibiotic, epicitracine, okay, and sulfamethyl-oxazole. Those are two different biotics that can differentiate species from different species under streptococcus. Okay, as we will see here, again, always refer to the table. So those are the two different antibiotics, okay? And you can see some streptococcus are sensitive to one, resistant to another. Some are resistant for both, okay? So this is a nice chart to give you an idea about the species that you are looking for. So in this case, what you see after incubation, here you don't need to worry about the, you need to look for the inhibition zone, okay? And you have two different antibiotic. You can see clearly very nice inhibition zone here. So your unknown is sensitive to this antibiotic. Here, there is no inhibition zone. Bacteria can grow happily around the antibiotic. So it means that they are resistant to this antibiotic. So again, you look for, first of all, hemolysis, and then you look for the those two different antibiotics. Okay, one important test, because Streptococcus and Enterococcus, they are sharing some similarity. Okay, and one way to be able to differentiate between the two is to use a media, we call it bile escalin slant agar. Bile escalin slant agar. This media, it has two things here. So we call it selective and differential. Okay, and mainly it is used to differentiate between two different genera. It separates Streptococcus from Enterococcus. How? If you have your bacteria is capable of growing, you need to look for growing first if they can grow. And the other thing that you need to look for is the ability to hydrolyze escholine. It's a substrate in the media that if the bacteria has this enzyme that can degrade it, it will hydrolyze it to produce glucose and escolitin. And that escolitin can be detected by if you add some reagent, it will produce this nice black color as you can see in this picture. So here in this media, you look first of all for growth. If they can grow, this is number one. Number two, if they are capable of hydrolyzing escholine. Why this test is important? Because here, if you go back to the table here, I have all of those are Streptococcus, different species of Streptococcus, and I have Enterococcus, two species from Enterococcus. So one way to be able to differentiate Strepto from Enterococcus is the ability to grow in 6.5 sodium chloride. We know staph can do that. Strep cannot. So strep are minus. They cannot grow in high salt concentration. Enterococcus can grow. So this is one test to differentiate between the two. The other test is the bile escholine again. So all species of Enterococcus, they can grow in the presence of bile and they can hydrolyze escholine. So you will see this kind of picture. Streptococcus, they vary. Some of them can grow in the presence of bile, okay? But like Streptococcus bovis, they can grow and also they can hydrolyze escholine. So how you differentiate bovis from Enterococcus? Salt. Those can grow in high salt, Streptococcus cannot. Okay. Some Streptococcus, like Streptococcus pneumoniae, for example, they cannot grow in the presence of bile and they cannot hydrolyze escholine. And this is for biogens, the very, very pathogenic variant of Streptococcus. They also do the same. So looking here, in this case, you look, this is uninoculated, this is inoculated. So you can see that this particular isolate can grow in the presence of bile, but they cannot hydrolyze escholine. So they are negative for escholine because you need to see this black in color to be positive. Okay. And this is a sodium salt. As I mentioned, one way to differentiate between Streptococcus and Enterococcus at growth at high salt concentration. So Streptococcus cannot grow. Enterococcus grow very well. Okay. So this is for Streptococcus. What you need to focus on. You need to see first of all the hemolysis. What type of hemolysis they do on the agar plate. Okay. Like in this case, this is a beta hemolytic. Those are very little tiny colony, and around the tiny colony, very nice clear zones of inhibition. And then look for the reaction to two different kinds of antibiotics. This is number 2. And then you will be able to differentiate between different species of Streptococcus. Don't take sodium chloride in consideration because sodium chloride will not tell you anything, because all cannot grow in this. So you look for the antibiotics and you look for the bile escalin media. This is for the strep. So now one big difference between staph and strep is the shape under the microscope. Those are clusters, those are chain. Strep are chain, but the same colony, the same shape, bucci. Number 2, their ability to grow in salt, high salt concentration. Haloguric. Staphylococcus, haloguric. Streptococcus, not. Okay, this is a big difference also between the two. The antibiotic used is they are not using novobicin. Novobicin used for Staphylococcus, but there are two different bacitracin and sulfamethoxazole used for Streptococcus. So now, any question for strep?
Yeah, Professor, I do have a question because I literally filled out the sheet as we were talking, looking at my
Unknown.
unknown,
Mm-hmm.
and it's not jiving because my optocin, I'm not sure if I'm saying that right.
I mean,
I know it's
you mean the other,
sensitive.
the third antibiotic?
Yeah, because there's only one option on here that shows it as sensitive,
Yes,
and mine
pneumonia.
is sensitive.
Mm-hmm.
However, it doesn't fit any of the other criteria as far as hemolysis, SXT, bile escalin, or
I mean, that's okay. If you feel like there's one test which is not agreeable, you know, with what you have, you might have so in your isolate was much closer to what? Which one, in your opinion? Based on those, like beta hemolytic, first of all, is it alpha, beta, or gamma?
Mine is beta complete.
So beta complete, so you are dealing basically in this area here.
Yeah, but
because it is not enterococcus. I didn't give you enterococcus, so you don't need to look for this.
Mm-hmm.
Okay, so you are mainly here.
But then as I go across the chart, it doesn't.
What about the bacitracin?
But the I don't know because I didn't see that. That's the one test I don't see called out in the handout.
You should. I mean, you should see that slide.
I don't.
You see the slide like this. You sure? Slide number 9.
I do, yes, I do see that, but I don't see any mention of. Yeah, mine doesn't say anything about.
Oh, so you don't have slide number 9? You mean?
No, mine just says after incubation, and then it shows opticin and SXT, and that's it.
Yeah. I mean, like a plate like this.
There is a plate like that, but it only shows opticin and SXT. I don't.
Yeah, there is no opticin. Yeah, we're talking about the bacitracin and this antibiotic here.
Because mine calls out it's P. is antibiotic, and it's in my handout. It says P equals opticin.
So what is the your isolate number again? Unknown number. Let me see.
Two.
Okay, let me go and see here what I have for strip number 2.
And then on the catalase, the picture is, and I have printed it out. I can look at it on the screen, but it is not real clear on whether it's catalase or not. I mean, I guess it's catalase, but I'm
Yeah, I
kind of
mean
guessing from the
it is
focus.
not at this point. I believe it is not an important differentiating test.
Hmm.
Okay, so you don't need to worry about this catalase now. But if you look at your number 9, you have the so yeah, that is the 2 different antibiotic here. It is including the optocine, which is P. Yeah.
P,
Okay, and if you look at the picture after incubation, you can say it is sensitive.
sensitive, clearly
Okay,
sensitive. Yes.
yeah. So if you go to this optocine sensitive, so you are in the pneumonia. But the problem with the hemolysis, you said hemolytic, beta hemolytic, right?
Yes, correct.
So it should be here. So we will go back to optocine, and the zone is 20 millimeter, remember?
Mhm.
So you need to look for if we consider 20 millimeter sensitive or resistant. The same deal with the novobicin. Remember the novobicin?
Yeah, and I thought you said anything above 17
But
millimeters
that
was
was for
sensitive.
novobicin. That was for novobicin.
Ah,
It's a different antibiotic. So you need to look for this particular antibiotic and
uh-huh.
see, zone 20 millimeter. Is it going to consider like in the negative range
Ah.
or on the positive range?
Oh, so those zones differentiate between the two?
Yes, yeah.
Okay,
Look at this and see.
got
Okay,
it.
and what else? Let me see. So you said it is
But
beta hemolytic. No, it is not beta hemolytic. It is clearly not beta hemolytic. Okay, because all those greenish colony. Let me share this with the other student and take their opinion. Okay. Uh, okay, can you see this? So this is one of the unknown from
yeah,
Streptococcus.
yep.
Anybody, can you see this? The other,
Yeah, I can see.
oh, so look at this here. So this is not a complete hemolysis. So you have partial hemolysis, and the colony is kind of greenish color. Okay, so
Okay.
now you are talking about alpha hemolytic.
Alpha. Okay.
Yeah,
Because I
so
was
that
looking at
that can that can change the whole thing. Yeah.
it looks like it's almost looks like beta where it's a golden halo, but
Yeah, but I mean,
that would look
really,
completely different.
beta is really, you know, the I was working with Streptococcus equi when I was doing diagnostics for horse strep infection.
Mhm.
The colony is so tiny, very very tiny, and the zone clearing zone around it is huge. So you can see one colony, and maybe the size of a dime around very small tiny colony, and the clearing zone around it is like a zone like the size of a dime. You imagine. So so tiny colony, and the clearing zone is tremendously big. Those are really pure beta hemolytic, very authentic beta hemolytic. If you see little lysis like this, yellow like that, not much, and the colony is kind of greenish a little bit color, okay, so you are dealing with the alpha hemolytic.
Yeah, and
Okay.
but one other question as I go across the chart, my bile is positive and escalin is negative, and I don't see anything that fits um, that criteria either.
So you can see they are growing, but
Yes.
they are not hydrolyzing escalin.
So
Okay,
I have a positive bile and negative escalin.
you have positive, negative, so you are. Um, what about let's see, agalactia is beta, and
Yeah.
so if you look at alpha. So you said positive and negative, right?
Yes.
Yeah, negative, negative, positive, positive, negative. Yeah, we'll look at this and give me some time and see exactly, if there's any of the test is not really making, not agreeable with the unknown, with the identification.
Hmm.
So yeah, let me see this and I will get back to you with this.
Okay.
So and it is definitely they are not growing in any salt, so they are not um, enterococcus, because I didn't give any enterococcus.
And then
Okay,
one last question. I don't mean to ask all the
no,
questions,
that's
but
okay. Go
I
ahead.
didn't see. I know you had shown me on slide 9 or so that bacitracin. I'm not sure if I'm saying that right,
Persistence, yeah, it
but
is not here. Yeah,
I don't see that on mine.
yes, it is not here. The
Okay.
persistence, and you see, uh, yeah, I see why. So
I mean, I could just put not tested, but because I don't see it there.
yeah, go ahead and do this. Just go ahead and put like it is not, I can see it. And as I mentioned, I need to have
Mhm.
some time, go through this in more detail and see, if there is, any test which is missing from this. Yeah, okay, there. But I mean here, the main thing, as I mentioned, you need to know those name of the media and the purpose of the media. When I give you in your test, in your practical, I'm going to give you like kind of more um, clear pictures, not going to confuse you with kind of a doubtful slide. So slides will be very clear. Okay. Some of those are, again, the pictures are nasty. This is what we have in source right now. So they don't look good at all, you know. But at least you can tell based on the figure, if they can grow in specific media or no. But I agree with you, there is a confusion here about the identification. So that was isolate number, you said number 2, right?
For strep was 2, yes.
Oh yeah, okay. I will need to see exactly uh, what is the best way to interpret isolate number 2.
Mhm. And when you ask us to do a report, it says should consist of 2 to 3 pages. Okay, that includes biochemical characteristics
Yeah,
of the
you said that
isolate.
this is isolate, the unknown.
Okay.
I identified this unknown as streptococcus, whatever.
Uh-huh.
Why? Based on the biochemical data that I was provided, with.
Uh-huh.
It
Okay.
was like this, like that, like that. If there is anything which is confusing or, not agreeable, you need to tell that, it
Mm-hmm.
that was, because remember, let me tell you this. If you look for, say, agalactia, streptococcus agalactia, okay, not every single species under for agalactia is giving the same kind of reaction when you grow them and bind esculin. See here, minus, plus, meaning that there are some variants that cannot grow on bile, cannot hydrolyze esculin. Some variant that can grow on bile and hydrolyze esculin. Some variant will be combination, So this is why you cannot depend on biochemical testing solely for identification. You cannot do that because not every single species will be the same. There are variants of those. Some mutations, some isolates, different environment can be different. So not every single biochemical test will be the same for all under the same species, never. It is hard to see this picture. So some variants will be there. This is what I mentioned. The most accurate way of identification, biochemical is not the standard way of identification. Biochemical testing will narrow down, and give you a hint, where you look for, what you look for. But DNA testing will be the ultimate, because they look for those specific sequence, ribosomal RNA sequence, and that will give you a very accurate picture about the identification. Okay, so the bottom line is, if you are dealing with an unknown and see some of the data is not agreeable, it doesn't mean that this is wrong. It means that you have a variant of that. So you need to find the most common feature that are there, and based on those you need to identify your unknown. So if, for example, my unknown is beta hemolytic, and it can grow in persistence, and it's sensitive to persistence and resistant to those antibiotics. But then, I said, oh, okay, it's a biogens. But then all of a sudden, I found them like growing in the presence of bile. So I said, oh, no, it is not biogen. No, don't say that, because they have very, very, like, all those are common. So you have 1, 2, 3, 4 tests agreeable with biogen, but only this test is not. So it doesn't mean that it is not biogen. It means that maybe it's a variant of biogen that has the ability to grow in the presence of bile and to hydrolyze esculin. You know what I mean? So this is always, you need to put this in consideration.
Professor, I just have a curious question. So a lab in a hospital, are they running some of these same tests, or do they have more sophisticated equipment to run these
Usually, yeah,
tests?
usually, okay, let me tell you this. If you are like in a hurry to know, like, if this strep infection, for example,
Yeah.
you don't need to take a sample from the throat and inoculate different type of media, and then like AP, I, specific for staph or strep, and wait for a couple of days to read. So there is always very, very rapid and quick way of doing this. Serology, serological testing. So you get the antigen, you get the bacteria from the throat, and they have specific surface antigen, and you have specific antibodies for those. So you do a very, very quick serological testing, serology, antigen antibody interaction, and in few minutes you will be able to tell if this is really, that particular unknown, an isolate. You don't need to wait a couple of days to identify it.
Mm-hmm.
If you say you have staph, okay, and you said, yes, I was able to identify Staphylococcus aureus, for example, which is highly pathogenic. Now you need to know what kind of antibiotic is affecting this. So now you need to grow. You need to isolate and grow and test different kind of antibiotics and see which one is the most effective. So that the whole thing can take maybe a couple of days or 3 days to be able to figure out antibiotic, sensitivity. So you need to administrate the right antibiotic to the patient.
So how are
It
they?
depends, it depends on how quickly you need the results. If you need it quickly, you can do serology, serological test, antigen antibody interaction. Quickly, you will identify it, but sometimes you need more details about, say, and susceptibility to different antibiotic, so it will take more time.
So how do they treat the patient between that 24 and 48 hours, trying to
Yeah,
figure out which
usually they give a
antibiotic?
very, very broad spectrum antibiotic.
Oh, okay.
An antibiotic which has broad spectrum, so they can mainly, and
Kind
or
of maintain it.
or a cocktail of antibiotics,
Mm-hmm.
till they get the confirmed, isolate that they need to treat.
Mm-hmm.
Yes. And most cases, their prediction is right. So giving a combination of antibiotics, you know, that help. They are not going to leave the patient for a couple of days without any treatment, waiting for the lab result. No, they can't do that. They can't afford this.
Mhm.
Yeah.
Yeah. Okay.
Okay. So, do you have any question for me? Anyone has any question for me?
No, are you posting any additional mastering microbiology chapters?
I did
You
for
did?
19 and 20, like a couple of days ago.
Okay.
Yeah. So always check your homework. Yeah. But they're not due this week. I think I extended the due date.
Okay, and so these two are due the 26th, and then we have the presentation PowerPoint deck that's due on the 24th, I believe.
Yes. Yeah.
And then,
Yeah.
okay.
But this is, you're not going to do any presentation. You're going to send me your PowerPoint presentation so I can post it for everyone.
Okay.
Okay. Okay, good. So we'll meet again on Monday and we'll go through some of the lectures.
Okay. All right.
Okay.
Thank you. Very helpful.
Professor, before you go, can I talk
Yes.
to you privately?
Yes, man. Yes, I can wait for you. No problem.
All right.
Okay, sure.
All right, I'll sign off.
It's okay. Thank you very much.
Okay, I think everybody has signed off. So,
Yeah, go ahead. Yeah, do you have any question for me? Go ahead, please.
Yes.
Koya?
Yes.
Yeah. How you pronounce your name, Koya?
You said it right. It's
Oh,
Koya.
okay, Koya. Okay. Yes, go ahead, please. I think
Yes, so I
we were emailing, because you have some concern regarding the identification, I believe, of the intro.
Yes.
Yes. Yeah. What is your concern? What is the problem?
So I wasn't sure what you was exactly talking about. Like maybe like when I read the instructions that I get a little confused.
Yeah.
I
So
think that's
give
what it
me
is.
some examples. What is the concern here?
Because you had said that you need to know the